The 1986 Chernobyl nuclear power plant tragedy turned the area to become the most radioactive on Earth, and now a worm that thrives in the rads may help human cancer studies. After Reactor 4 melted down, humans were evacuated, but plants and animals survived the high radiation levels. A new study discovered that certain animals in the Chernobyl Exclusion Zone are morphologically and genetically distinct from others, raising issues about how persistent radiation affects DNA. New York University researchers found that tiny worms living there now have developed highly tolerant to chronic radiation from Chernobyl. The discovery may explain why some people with a hereditary susceptibility to cancer get it. Chernobyl was a tragedy of incomprehensible scale, but we still don't have a great grasp on the effects of the disaster on local populations, said Sophia Tintori, NYU's postdoctoral associate in biology and the study's first author in the Proceedings of the National Academy of Sciences. Did the sudden environmental shift select for species or even individuals within a species that are naturally more ionizing radiation resistant? Tintori and her colleagues used nematodes, microscopic worms with simple genomes and quick reproduction, to study basic biological processes. These worms live everywhere and they live quickly, so they go through dozens of generations of evolution, while a typical vertebrate is still putting on its shoes, said NYU biology professor and study senior author Matthew Rockman. I had seen footage of the exclusion zone and was surprised by how lush and overgrown it looked. I'd never thought of it as teeming with life, said Tintori. If I want to find radiation-tolerant worms, this landscape may have selected for that. Tintori and Rockman visited the Chernobyl Exclusion Zone in 2019 with Ukrainian and U.S. scientists, including biologist Timothy Mousseau of the University of South Carolina, who studies radiation from the disasters. They collected worms from dirt, rotten fruit, and other organic material, using Geiger counters to monitor radiation and PPE to protect against radioactive dust. Worms were obtained from areas with radiation levels ranging from negligible, New York City, to high, space, harmful for humans but unknown for worms. The crew took samples from the field to Mousseau's field lab in a former Chernobyl residential home to isolate hundreds of nematodes from soil or fruit. They then used travel microscopes to isolate and cultivate each worm in a Kyiv hotel. Researchers froze the worms in the NYU lab to investigate them. We can cryopreserve worms and defrost them for research. That implies we can pause evolution in the lab, which is unachievable with most animal models and useful for comparing creatures with diverse evolutionary histories, said Rockman. Genetic and evolutionary research have employed Oscaeus tipuli worms, therefore they examined 15 of them. They sequenced the genomes of 15 Chernobyl O. tipuli worms and compared them to 5 from other countries. The researchers were startled to find no radiation damage signal in Chernobyl worm genomes after repeated studies. Tintori said, "This doesn't mean that Chernobyl is safe. It more likely means that nematodes are really resilient animals and can withstand extreme conditions. We also don't know how long each of the worms we collected was in the zone, so we can't determine its ancestors' exposure over the past 4 decades." The researchers designed a system to compare how quickly populations of worms grow and measured how sensitive the descendants of each of the 20 genetically distinct worms were to different types of DNA damage to determine if the lack of genetic signature was due to Chernobyl's worms' unusually effective DNA protection or repair. While the lineages of worms were different in how well they tolerated DNA damage, these differences didn't correspond to the levels of radiation at each collection site, so unlike the origin stories of several superheroes, radiation exposure doesn't seem to create superworms, just as it can't turn you or I into Spider-Man or the Hulk. Instead, the teams found that Chernobyl worms are not inherently more radiation tolerant and that the radioactive terrain has not driven them to adapt. Despite O. tipule's genetic simplicity, the findings suggest that DNA repair can vary by individual and may help researchers comprehend human natural diversity. Now that we know which strains of O. tipuli are more sensitive or more tolerant to DNA damage, we can use them to study why some people are more susceptible to carcinogens, said Tintori. Cancer researchers want to know how various animals respond to DNA damage to understand why some people with a hereditary susceptibility to cancer get it. Thinking about how individuals respond differently to DNA-damaging agents in the environment will help us understand our risk factors, said Tintori.